what about synapses? We've talked about two types of synapses. I'll remind you that those two types of synapses include electrical synapses in which there's an actual physical connection, a protein with a pore through the middle of it that connects two neurons, actually multiple ones. These are gap junction proteins, and the connections are called gap junctions. The pore through a gap junction is big enough to allow ions and small molecules to get through. So if there's an action potential coming down here, remember, here's our voltage-gated sodium channel, action potential coming down, allowing sodium into the cell. Okay, when it gets to the electrical synapse, that sodium can go through the gap junction and start a new action potential. So here's a voltage-gated sodium channel in the next cell, and if you get enough charge there to get it to threshold, that pops open, and that's an electrical synapse. They're not all that common, but they're especially important wherever there's selection favoring very fast transmission with no particular ability to adjust it. These are not very adjustable. Chemical synapses are more common. They're slower, but they're adjustable. Which makes the adjustment, the ability to adjust them, is pretty important for most animals. So I just drew one when I was talking about action potentials, but let's run through it again. So we've got the presynaptic cell and the postsynaptic. And the synapse itself right in between. Okay, in a chemical synapse, again, you'd have voltage-gated sodium channels opening up as the action potential moves along. And that sodium, that positive charge, would open voltage-gated calcium channels, allowing calcium in. That calcium interacts with existing vesicles by binding to a calcium binding protein. That calcium binding protein, once it's bound to a vesicle of neurotransmitter, that allows the neurotransmitter out. The neurotransmitter can bind to a receptor. In this case, I'll show you a metabotropic receptor. Interacts with, for example, could be a G protein that might activate adenylate cyclase to produce cyclic AMP, which might open a channel by binding to it and affecting whether or not ions can enter the cell. And if enough ions come in, you get a new action potential. Okay, so as part of our toolkit to understand how information gets passed around from one animal to another, electrical synapses, fast but not flexible. They're hard to adjust. Chemical synapses are flexible. You can adjust the amount of neurotransmitter. You can adjust the availability of calcium. You can adjust the number of receptors. So both the presynaptic and the postsynaptic cell can adjust. They can adjust the amount of secondary messenger available by adjusting the amount of the enzymes that make them or the channels that open them. So chemical synapses are very adjustable, and animals use them to adjust their responses. That's going to be part of the neural circuits that we're using in this class.